And we are alive. Hello, everyone. Good afternoon. It's a pleasure to be here with the researcher Vanessa Matta. I'm going to read the biography from Vanessa so you know a little bit more about her work before the talk. Vanessa is a researcher at the Research Center in Biodiversity and Genetic Resources from the University of Porto, Portugal. Her work has mainly focused on the use of molecular tools, in particular metabarcoding, for the study of species interactions, community ecology, and ecosystem service. She's currently working as a postdoc under the project BatPine, promoting the resilience of ecosystem service to climate change, a case study with pine plantations, pine processionary, moths, and bats. She was recently awarded a 6-year junior research position at CIBIO InBIO. It's a research center in Portugal. She's also vice leader of a cost action working group looking at the effects of climate change on the provision of ecosystem service by bats in Europe. And although mostly she works with birds and bats, she's also a very enthusiastic field ecologist, and she collaborates and works with many other different projects, including insects, reptiles, fish, and other mammals as well. It's a very big profile for such a young researcher. So it's a big pleasure to have here Vanessa Matta, and she'll be talking about when community ecology meets genetics, metabarcoding, its never-ending applications. So with you, Vanessa Matta.
Hello, everyone. So thank you, Hernani, for your very nice introduction. So as Hernani said, I'm a postdoc researcher working at CIBIO, and I would like to first thank Hernani also for this opportunity to be here talking with you about my work on metabarcoding, and hopefully it won't get too technical, but I'll try my best. So first of all, for those of you who have never heard about CIBIO, which is most of you, I guess, it is a research center within the University of Porto. It is located close to Vila do Conde, which is a small town north of Porto, and which is the second largest city in Portugal. So the center itself is located basically in the middle of nowhere, surrounded by cornfields and eucalyptus plantations. But if we zoom close enough, you can see that we have a swimming pool. So what more could we ask for, right? We are about 350 people, about which half are PhD and master students of CIBIO's BioDiv program. Many of our researchers and students are not Portuguese, which means that we have a very diverse and international environment in our center, with people working on very different subjects, from adaptation of animals to desert environments, to genomics of coloration, and biomechanics of scorpions, for example. And the other good thing also in our center is that we have very nice lab facilities where researchers, lab technicians, and students can do most of their molecular work. So currently, I'm a member of 2 research groups, one focused on bat ecology and the other on general aspects of applied ecology. And a lot of the things that I will be presenting to you now will be, of course, done in collaboration with all these people, each doing their their part and contributing to the work. In particular, here Gary McCracken and Martin Corley, although they are not really CIBIO members, but they have been collaborating in a lot of these studies. So thank to them also. So I entered CIBIO as a master student, and for my master thesis, I was working with bird blood parasites and trying to compare bird parasite communities between Northwest Iberia and Northwest Africa. And at the time, we found some nice results, for example, in which parasite communities seem to be shaped mostly by the bird community itself, but also that host-specific lineages were less likely to occur across the Strait of Gibraltar. And unlike these, the host generalists were mostly cosmopolite, so they were occurring everywhere. And another thing we found, because we were using 3 slightly different but overlapping primer sets, is that individuals often harbored more than one parasite lineage. So a lot of the birds had 2 or 3 or 4 or many different parasites. And while in most cases we could detect these double infections, let's say we would have a very nice chromatogram and then suddenly you have 2 peaks in some positions, which indicated this double infection. When things were too different or too many lineages were present, things became a little bit chaotic, and it was impossible at the time to disentangle these mixed infections. So in 2013, when I was given the chance to work with free-tailed bats and my supervisors were interested in analyzing its diet, the idea at the time would be also to look at these chromatograms resulting from Sanger sequencing and trying to disentangle the different pre-item sequences. I thought, no way, this is going to be impossible and we're not going to be able to differentiate different insects inside the chromatogram. That's crazy. So I started reading as much as I could find on molecular diet analysis. And although at the time the literature was not that extensive as it is today, there were already a couple of papers on bat diet, especially from Elizabeth Clare. At the same time, our institute had just received an Illumina platform, so when I proposed we try this metabarcoding approach, we all thought that probably this was the best idea to try to move forward. So to present you our first study, I would like to give you a little background information on this species, the European free-tailed bat. They are mostly distributed around the Mediterranean basin, and they are fast flyers, flying up to 130 kilometers per hour. So they have these very long and pointy wings to forage at open spaces, usually around 200 to 300 meters high, but they can also go up to 1,600 meters, which is really high for a mammal. So from the literature, what we knew was that they were supposed to be moth specialists, sometimes feeding also on Neuroptera. However, lack of taxonomic resolution in the analysis of moth fragments present in feces due to their soft body and lack of taxonomic characters had until now prevented any further analysis. So our goals were to understand if females and males were eating the same, but also if juveniles were eating different things from adults, and if the diet changed throughout the breeding season. So we captured the free-tailed bats in these very large and high bridges using this rudimentary setup of nets in which we had these strong metal poles falling down from the bridge, and then we have the nets here. So when the bats left the roost, which is located along some cracks along the bridge, you can see here the bats hiding. When they leave the roost, they get trapped in the net, and then we can pull the nets up to remove the individuals. So we tag the individuals, so we would know which bat is which. We also sex them, we age them. Unfortunately, we didn't collect any wing measurements that we could use, for example, for geometric morphometrics. But that's something that would be very interesting to do, also to correlate with the diets. So for the metabarcoding analysis, we pretty much followed what was being done at the time. So we amplified a small CO1 fragment using the ZEAL primers, and we extracted DNA from a single pellet of each individual. In total, we had 143 individuals distributed across 5 roosts, and the samples were collected between April and October. So what did we get? As well as what was known from the literature, we found that they were indeed feeding mostly on Lepidoptera, so this big yellow section over here. But we found in total 5 different orders of insects. So they were also eating some Coleoptera, Diptera, Hemiptera, and Neuroptera, which were distributed across 17 different families and over 100 different insect species. We then tried to assess if the prey composition varied among our predefined groups. And what you can see here is that we found significant differences in prey composition between males and females, but not seasons or age categories. We then tried to understand which prey items were causing these differences. And so here in this plot, you can see the frequency of occurrence, so the percentage of samples in which a prey was found on both males, here on this side in black, and females on this side in white. And what you can see is that females seem to be eating a lot of these moths over here, while males seem to be foraging more on these moths over here. And what we can kind of say from these moths at this point is that they seem to be larger species and of migratory behavior, while these ones tend to be smaller and non-migratory. So we wanted to test this further, and we plotted and tested the wingspan of each moth species and also its migratory behavior. And what you can see is that females are eating larger prey than males, but also that they're eating a higher proportion of migratory moths than males. So we found gender-related segregation in the diet of these bats. This is mostly likely linked to the high energetic costs of pregnancy and lactation of females. And what we hypothesized is that females are probably hunting and foraging at higher altitudes, taking advantage of migratory moths, which are rich in lipids for their migration, and thus highly profitable prey. Furthermore, we could only detect these patterns because of the high resolution offered by metabarcoding and the fact that most moth species in Europe have been sequenced, and we could thus infer their size and their behavior. So, after this study, we were very happy, but we also started having a lot of questions. So we started wondering how good was our sampling? Was one pellet per individual enough? Would 5, 10, 15 different pellets of the same individual tell us the same thing? What if we pooled pellets instead of analyzing them individual before doing DNA extraction? We couldn't find any information on the effects of sampling procedure in molecular dietary analysis, so we decided to explore these in more detail. So we came up with an experimental design where we sampled 20 bats, bats that had pooped a lot, so they had left over 30 different pellets in a single foraging night. And we extracted DNA from 15 individual pellets, and we extracted DNA from a pool of 15 pellets. So we had 16 samples per bat. And we were also interested in understanding the effect of technical replicates. So we also did 3 PCR replicates for each of these samples. So what we found is that most of the variation in diet composition comes from individuals. So you can see here the highest R-square is for individual, and also the highest mean sum of squares is from individual, followed by pellets, and then replicate. And in fact, you can see that the mean sum of square of individuals is 15 times higher than that of pellets, which is in turn 12 times higher than that of replicates. So it seems that really individuals are the ones with most variation, and then you have some variation within the pellets, and then a little bit variation, but very little between PCR replicates. We also conducted a bunch of simulations in which we randomly subsampled the number of pellets that we analyzed. So here you can see the number of pellets used. For example, if we take 1 out of the 15 pellets analyzed, what would be the frequency of occurrence of these moth species? And what if we analyzed 2 and 3 and 4 and so on? And we generated these curves, the simulated curves, and each circle here represents the proportion of times that a certain frequency of occurrence was observed. And you can see that as we increase the number of pellets analyzed, the frequency of occurrence converges to the real observed frequency in the 15 pellets. And here you can see the frequency error, of course, decreases with the number of pellets. But also what we can see is that it also depends of the frequency of occurrence within pellets, so within individuals. So we'll have some moth species, for example, that will be detected only after analyzing a very large number of pellets, while others will be detected very rapidly, and with just a couple of pellets, we'll reach the true observed frequency of occurrence. What about pools? What happens when we pool the samples before extracting the DNA? So here you can see the average number of species per sample in summing the 15 pellets, analyzing each pellet individually, and pooling the 15 pellets. And what you can see here is that the average number of species that we observe in a pool is pretty similar to the average species that you observe in a random single pellet, which is much lower than what happens when you sum up the 15 individual pellets. And at the time we were a bit confused of why this was happening, and we thought, okay, maybe this has to do with sequencing depth. Maybe we are not generating enough reads to capture the entire diversity. So we repeated the experiment, this time with more reads, with higher coverage, and what you see is exactly the same. So the number of species detected in a pool is very similar to the number of species detected in a random single pellet. So we went a bit further and tried to understand what exactly were we detecting in these pools. And here in the x-axis you have the frequency of occurrence within individuals and the probability of detection in pool. And what you can see is that for prey items that occur in all the 15 pellets, there's a high chance it will be detected in the pool. But if for some reason the species is only present in 2 or 3 or 4 pellets, then the probability of detection in the pool is much lower. And this makes sense, of course. So the detectability in the pools depends on the frequency of occurrence within individuals. Still, if we assume that the 15 individual pellets correspond to the truth, so these here being the truth, we can see, these here being the pools, we can see that even for very frequent items, so items that are present in over 50% of the individuals, when you analyze multiple pellets, your frequency error goes down. And if you analyze just one, you see that it's similar to that of pools. So the error in estimating the frequency of occurrence in your population, in your sample of individuals, is similar between using a pool of pellets or using just one random pellet. So to wrap up, individuals have most of the variation in diet. More pellets give more information, so more prey species about each individual. And the population frequency of occurrence error decreases with the number of pellets used and, of course, the frequency of occurrence within the individuals themselves. And finally, pooling samples or analyzing just one random seems to be equal. And of course, this detection in the pool will depend on the species frequency of occurrence within individuals. So, like before, we felt like we had answered some of our methodological questions, but because we were interested in working not only with strictly insectivorous species, we wondered also what would happen if we tried to study the diet of a generalist species. So we would probably need to use multiple primers for the different components of the diet, for example, insects, plants, and others. but we were not quite sure how the different results would be among the different primers in terms of prey diversity and composition, as well as if our molecular approach would miss or overrepresent some prey items in comparison to traditional morphological analysis. So as a model system, we use the black wheatear, Oenanthe loucura. Just to give you a bit of background on this species, it is a highly territorial species in which males actively defend their territory, kicking out any intruders. They also spend a lot of time singing, signaling to others that this is their space. It is mainly an insectivore, but can sometimes also feed on wild berries and small reptiles. So we captured with spring traps over 100 individuals and collected their droppings. And we then used 4 different molecular markers targeting different genes for the different prey items. So we used 18S to amplify the entire eukaryotes, so plants, invertebrates, vertebrates, and whatever else showed up. TRNL, which would be amplifying a small fragment of plants, CVJ from CO1 for invertebrates, and this primer here, 16S, focusing on the 16S gene, also for invertebrates. And we also did morphological identification of the prey remains to compare all these different approaches. We also tried, when we analyzed the diet, we considered both each molecular marker individual, but also tried to combine them all together and see how different these would be. So looking at plants first, we can see here that overall we detected a much higher number of occurrences using molecular techniques. Most of these occurrences were of taxa identified at family level, while using morphology, many of the taxa were actually identified here at the species level. This was expected as the molecular markers used do not usually show very high taxonomic resolution. What we can also see is that the molecular methods detected a much higher diversity here of plant taxa than did the morphological analysis. So we have many different plant families here, some with very high frequency of occurrence that were not detected at all by looking at the remains. So taking into account what we know about the behavior of the species, it is almost impossible that these birds are eating all these different plants, as they are known to eat mostly berries, and most of these plants do not actually produce any fleshy fruits. So most of the plants detected here are actually most likely the result of secondary detection, in which case the plants were consumed by the insects that the birds ate. So now moving on to animals, the overall findings were fairly similar with a few important differences. We can see that this time using morphology, a lot of the identifications were done at either family or order or even higher level, while of course metabarcoding was able to identify a lot of things at species level, sometimes genus, and sometimes family also. We can see also that there seem to be some differences between the different primers. If we focus over here, for example, we can see in blue the CIBIO is detecting a lot of Diptera and Lepidoptera, while the other 2 primers in orange and yellow do not seem to be detecting those insects so much. And on the contrary, for example, for ants, for example, here, you can see that the orange and yellow have very high frequency of occurrence, while CIBIO, the blue, is really over here. It also, if we also compare the morphology and the molecular approaches, we can see that this time there seems to be an overall higher congruence. So we have a lot of detections of ants using both morphology and metabarcoding. The same here for Hemiptera, also some spiders over here. But then there are some groups which are really not congruent at all, particular Lepidoptera and Diptera, which almost do not occur when looking at morphology, but seems to occur a lot using the ZBJ. So in this case, it seems that we have a problem, let's say, of primer bias, in which this ZBJ is detecting really residual amounts of DNA of Lepidoptera and Diptera, probably traces of past meals that are no longer detectable using morphological analysis, and that even the other primers did not detect because they had higher quantities of other things to amplify. So to wrap up, metabarcoding is powerful, but for generalist species, single primers can give biased views of the diet, so we can have issues of primer bias. And we also need to make careful interpretation of dietary items because there can be cases of secondary detection. Now, as we found these cool differences between males and females for bats, we were also interested in checking if the same thing happened with these birds. And another thing we were interested is whether these differences would only be detected if the analyses were done at the species or OTU level, disappearing at family or order level, which is what we usually have with morphology. And in fact, what we found here is that males are having a wider prey richness than females at prey items, so species or OTU level. And this is likely related to their higher mobility with, when they're performing territory defense. On the other hand, if analyses were done at the family level, you can see that these differences do not exist anymore, so they are feeding on a similar range of prey families, and this is what you would find if you were doing morphology. So if you were analyzing the diet of these birds, looking only at morphology, you would say males and females are eating the same thing when in fact they might not be. What they are seen to be doing is feeding on a similar range of prey, but in terms of functional diversity. So they're eating the same type of prey, but they are eating, one gender is eating more items than the other. We also tested for differences in diet composition, and again, we found differences between males and females, this time not only in terms of species and OTUs, but also of families. So here we plotted the frequency of occurrence of each family item, and the ones in green are the ones that are significantly different between males and females. So each line represents an interaction between a female and a certain prey family, and in this up here between males and a certain prey family. And actually, the prey item that mostly differed between sexes was ants. So for Mycidae here, in which females were 58% of the females were eating ants, while only 29% of the males were doing so. And this is likely related to females foraging closer to the nest, while males spend most of their time moving and defending their territory, with ants being a highly predictable and abundant and easy to catch prey for the female, while males are eating more random things around their territory, things that they can find while they're moving around, which also explains the higher diversity in terms of prey species. So now moving on into something a little bit different, which is trying to use metabarcoding to understand pest control services by flying vertebrates. So it's more of a applied problem to agricultural and forestry systems. So as you might know, birds and bats can be quite widespread in agroforest landscapes, and many studies have pointed to their overall negative effects on the abundance of arthropods across multiple agricultural systems. However, one thing that is often not clear with these studies is how individual species contribute to this service, nor how it varies seasonally, which can have important consequences on management and conservation decisions. A way to disentangle this is by the means of ecological networks. So by studying species position within the networks, we can get a deeper insight into the ecological role of species and their functional role in ecosystems. For example, by looking at the modularity within the network, we can see how many units the provided service is divided, or by looking at the centrality of the species within the network, we can infer its importance in the community. So combined with powerful tools like DNA metabarcoding that can offer species-level resolution to dietary items, ecological networks can provide a key understanding of species roles in the provision of ecosystem services. So now I'm presenting to you two practical case studies on how we can use ecological networks and metabarcoding to better understand predator-pest interactions. One of the studies focuses on bats, while the other focuses on a community of birds. And so for bats, we conducted a spatial and temporal restricted sampling in order to identify the roles that different species play in pest consumption. We wanted not only to describe the overall predation on arthropod pests, but also produce a network and assess the species' role in the network. Finally, we wanted to understand the functional complementarity and redundancy across species in order to identify key species for the service. Regarding birds, we conducted a seasonal sampling, so every month, but in a spatially restricted area, this time in order to describe seasonal variation in the predation of arthropod pests and produce seasonal predator-pest networks and describe the seasonal changes in network properties. So for bats, sampling was made here, northeast Portugal, within the Subwater River Basin, using mostly mist net trapping. We captured almost 500 different individuals of bats belonging to 19 different species, and we collected over also, so summing to these 400 individuals, we collected also over 100 fecal pellets from known roosts, and we extracted DNA now using previous knowledge. So we extracted 3 pellets individually from each bat, and we analyzed over 1,200 droppings. So we had over 500 samples, and we had 3 pellets, or 2 or 1, depending on the individual, pellets per individual. We then used 2 CO1 primer sets to amplify the insect DNA present in the feces. So we used ZBJ, which has all these biases problems. So we also included a different CO1 primer, which is this FWH, which is more generalist, is able to amplify a wider range of prey. Regarding birds, sampling was also conducted in northeast Portugal, but at the catchment area of the Tua River. So you can see here, this is the Tua River and this is the Douro River, and birds were captured here and here. Birds were captured also with mist nets once per month during 13 months at these 4 sites. In total, we sampled over 50 different bird species, and over 2,600 droppings were collected and its DNA extracted. So this time we used only CO1 primer because from the knowledge that we gained with the bats, we could see that FWH2 actually represents most of the diversity, and that we don't gain anything by using the two primers. So now moving on to the results regarding bats. Overall, we found that pest predation levels differed among bat species. So some species have most of the individuals consuming at least one pest species, while other bats almost predate no pest insects at all. Bats in our community predated on at least 132 different pest species belonging to 47 families and 8 insect orders. So Tipula oleracea was the most predated pest, followed by noctuids, and finally also Tametopeia pithiocampa, which is the pine processionary moth, and also Praise olei, which is the olive moth. And this makes sense because our landscape has a lot of both pine trees and olive trees, so these pests that showed up, they make sense. So now moving on to the network. Our bat-pest interaction network was modular, with bats being clustered into 6 different groups. So each group here represented with different colors. You can also see them here in a matrix shape. Each module or cluster was associated to pests belonging to different orders of insects, whose frequency of interaction varied according to the model. So, for example, this purple model here interacted almost exclusively with Lepidoptera. This purple here also mostly with Lepidoptera, but then other clusters interacted with other things. So this yellow one interacted a lot with Orthoptera, while this red one with Diptera, and so on. Of course, they all interacted with moths because bats predate a lot on moths, but you see that there are really some differences on the prey items that each cluster is interacting with. Now, regarding the species roles in the network, most of the species were peripherals, meaning that they were neither connecting the different models nor contributing to the cohesion of the models or the overall network. Still, for example, Miniopterus schreibersi, which is one of the species, and Rhinolophus eposiderus were identified as model hubs, meaning that each is a strong representative of its cluster. Yet, the species with highest centrality scores were Miniopterus schreibersi, Rhinolophus ferrumecino, and here Pipistrellus pipistrellus, exhibiting not only the higher number of connections or prey, but also a higher proximity to all the other bat species and the higher connecting role to the different models. You can actually see here in the network how that they do show central position. So they're not here at the edges. They're actually here in the middle, interacting with a lot of different species and with species of other models. Also, network nestedness, so how the diet of specialist species is contained or not within that of generalist species, was lower than expected. And this means that the specialist species had a distinct diet from those of generalists. So they were eating different things and not really a subset of what the generalist species were eating. Finally, we simulated bat extinctions in order to see how the number of regulated pest species would drop. So each triangle here represents the percentage of pest species that is being regulated by bats according to the number of bats that you extinct. So if you extinct 5 bats, how many pests are still being regulated? This is what the curve is showing us. And this curve here is extincting bats according to their abundance, or not to their abundance, but to the number of interactions that they show. So the last bat being going extinct is the one that shows a higher number of interactions in the network. And you can see that this curve here is quite smooth, so it takes a long time for the number of regulated pest species to drop. And this means that bat species show a high level of functional redundance among them. And in fact, 60% of the pests can be regulated with only 3 bat species. So our 3 top ones are these species here. Pipistrellus pipistrellus, which is very common, Rhinolophus ferromechinum, not so common, but still fairly, and foraging mostly on forests, same with Rhinolophus oreale. You can see actually that these 2 species here, their triangles are yellow and red, which means that their conservation status is, if I go back, they are vulnerable or critically endangered, so they are protected species of conservation concern. This percentage of regulated species can go up to 75% if we include 3 more species, so Miniopteros, Plecotus, and Tadarida. However, this scenario of extincting bats according to how many interactions they show with pests is not very likely. It's very unlikely, actually. A species would go extinct depending on how useful they are. A more realistic scenario would be to extinct species according to their conservation status. And that's what I'm going to show you now. So these squares here are species going extinct randomly, so with no order whatsoever, while this bottom curve here is bats going extinct according to their conservation status. So first extincting, critically endangered, then vulnerable, and so on. And what you can see here is that although not very strong, it is still significantly different. There's a reduction, there's an accelerating decline in the percentage of species being regulated if bats go extinct according to their conservation status. And in that case, we need almost 3 times more species in order to also regulate the same number, the 60% of the pests. So now moving on to birds. Pest predation levels in birds were much more even across the different species what we observed than what we observed with bats. On average, birds interacted with pests around 55% of the times, and the number of positive samples, so positive to pest species, seems to be more a response to the number of samples tested. So here we plotted the number of samples per bird species and the number of samples with pests. And you can see that there's a very nice correlation between the number of samples that have pests and the number of samples tested. So it seems that the bird species is not really an explaining factor in this case. Still, there are 2 species here which seem to be slightly above average. So this is a swift and this is a small warbler, and for some reason they seem to be interacting a little bit more with pests than the remaining species. Now, if we look at the most consumed insect pests, we can see that there's a strong seasonal variation associated with each pest species, some being predated, for example, only here in autumn, while others are showing different peaks. So this one peaks here in September, while this one peaks in November. These seem to occur mostly in winter and early spring, and so on. And this, of course, has likely to do with insect phenology. So, in fact, this pattern seems to expand to the entire community, not only these very common insects. So we plotted our pest predation into an MDS. So each point here represents a site and a sampling month. And you can see if we if we plot things through time, so first month, second month, third month, fourth month, and so on, that there seems to be a cyclical pattern going on. So there's a cyclical and gradual shift in pest consumption throughout the entire sampled year. So this is April, and this is the last sampled April. So the communities go around and then return to the same position. And this is likely related to insect phenology, with some species starting to appear, then disappearing, but others starting to appear and then disappearing, and so on until you complete an entire animal cycle. Finally, if we take a look at the network metrics associated with bird-pest interactions, we can also observe a strong seasonal variation in the metrics. For example, we have modularity being highest in spring, here spring, and being lowest in autumn and winter, indicating a much more homogeneous community that mostly shares resources. We can also observe that there was a peak in pest richness, for example, here in autumn, that was not accompanied by a higher bird richness. So these led to an overall higher niche overlap among species and also a drop in specialization and modularity. So we have a higher pest richness here, but we have the same number of birds in our community. So we have a higher niche overlap over here and a lower specialization level. So to conclude, DNA metabarcoding combined with ecological networks constitute a valuable framework to study pest control services by insectivorous vertebrates, allowing not only to identify potential candidate target species for conservation biocontrol, but also allowing a deeper understanding of predator-pest interactions and dynamics. So with that, I'm done with my presentation. I'm happy to take questions next. So make sure you follow the link and we can chat a bit. But I'm also happy to be contacted by email if you have any further questions or would like to discuss some ideas or topics on future work. And that's it.